Hi all, welcome to the session on Insilico drug repurposing.
Drug repurposing is also called as drug repositioning, reprofiling, or retasking. It is a strategy for identifying new uses for approved or investigational drugs that are outside the scope of original medical indication. This strategy offers various advantages over developing an entirely new drug for a given indication. So now if we have any pandemic diseases or epidemic diseases which are spread worldwide, like the corona which is happening in the present, the need of a drug is very quick and the drug has to be discovered very immediately. So in those cases we will be using drug repurposing. So the failure of the drug will be minimal and also we can discover the drug in a very short period of time. So what are the benefits of drug repurposing is, it is fast, cheap and good for very rare diseases. Also it can be used for epidemic and pandemic diseases, and the drugs that are used for this process are already known drugs, so the safety profile and side effects are already known and the history to how to use it with humans is already known. So it reduces the requirement of clinical trials, that is the first phase and the second phase. So it saves a lot of money and time for us, and also the pathways of action of these drugs are also known, which can help in repurposing process. So first and perhaps the most importantly, the risk of failure is lower because the drug repurposing has already been found to be sufficiently safe in clinical models and humans if early stages of trials have been completed. It is less likely to fail, at least from a safety point of view, in subsequent efficacy trials. Second, the time frame of drug development can be reduced because most of the preclinical testing, safety assessment and in some cases formulation development will already have been completed. The third and the last point on drug repurposing is, less investment is needed, although this will vary greatly depending on the stage and process of development of the repurposing candidate. So with these three main advantages of drug repurposing, we will see what drug repurposing involves. So the different types of drug repurposing are knowledge-based drug repurposing and activity-based drug repurposing. Knowledge-based and activity-based drug repurposing you have already seen in the first part of the module. So some of the in silico drug repurposing tools that have been used in today's world are, first, molecular docking techniques, transcriptional signatures, then network analysis, data mining, machine learning, similarity analysis, from where we go to retrospective studies for validation and then meta-analysis, and then we can then validate these in silico results with our in vitro analysis like cell culture studies and protein signatures, and also the in vivo analysis, xenografts, and directly take it to clinical trials. In clinical trials we do not need phase 1 or phase 2, we can directly go to the phase 3, or sometimes it can start directly from phase 2. So this will skip some time and the time period will get reduced. So repositioning on the rise, big pharma companies have taken this compound library and even academic institutions, and they are funding the academic institutions to do drug repurposing. So in the early stage of repositioning, academic institutes have been funded for it, after which the late stage of development, approval and marketing happens, after which the old drug can be used for a new purpose, and again the traditional marketing is done by the big pharma companies. So it is a cyclic process from where the old drug is taken from the pharma companies by academic institutions and the funds are also given, and then repositioning is done, after which the old drug is given for a new treatment process, and then it is again traditionally marketed by the pharma companies. It starts from the pharma companies and then it ends back with the pharma companies. So this is a cyclic process. So a traditional drug discovery is a very long process which takes up to 20 years, but drug repurposing will reduce it in a very shorter span of time. So the difference between traditional drug development and drug repositioning is as follows. In drug development, we will identify the therapeutic target and develop potential drug candidates, design it and then synthesize it and then test for cell culture assays and then take it for animal testing, from where the drug, after which once the drug gets successful in the laboratory, which itself is a very big task, most of the drugs fail in the laboratory itself, and then it is taken for clinical trials, and then there are many chances of the clinical trials getting failed. Phase 1, phase 2 and phase 3. Only when it passes all these, the drugs are being approved for clinical uses. So this is a very long process. But whereas in repositioning, if we can see, the identified drugs are all directly taken to phase 2, as I've already said, and if they are effective in phase 2, it is taken to phase 3 and drugs are reapproved for new uses. So if you can see, the chart is a very long path in traditional drug development, whereas in drug repositioning is a very short but effective method. So again, another comparison in terms of time. So traditional drug redesigning takes around 10 to 17 years, whereas repurposing takes a maximum of only 8 years, which is even lesser than the minimum period of traditional drug design. So as we have seen process, the success rate is less than 10% in traditional, but whereas in drug repurposing we have reduced safety concern because the drugs are already known for its safety and its side effects. So the chances of failure are very less, and also it can be marketed quickly. So the drugs that have been used in the previous years and which have got repurposed are mentioned here. So first, if you can see, amphotericin, which have been given for fungal infections in the previous years, but now it has been repurposed for leishmaniasis. So for example, if we take a drug and it has been repurposed, so there can be two reasons for the drug to be repurposed. One is when they have side effects, it can be repurposed for the other side effect diseases. Second is when even though the drug has a better activity to the first disease itself, it can still be given to another disease. For example, if you can take thalidomide, first it was given for morning sickness, which caused patients to be which caused babies to be born without arms and legs, but later because of its side effects it was directed and used for leprosy. Same way, these are the other drugs that were used. So first, in the earlier purpose it was being used for these conditions, and then next it was used for repurposed condition. So rosiglitazone was first given for diabetes, but then it was repurposed for malaria. So these are some of the examples that we can use for a drug repurposing approach.
Thank you for watching the video.